Buongiorno. I'd like to thank the organising committee for asking me to speak. It's quite early, and there's a little bit of genetics in this, so I hope we can follow through together. So it's the title of the talk is Developing a Gene Therapy for Aniridia-Related Keratopathy, and I'd just like to present some very early data that we have to try and tackle this corneal disorder. So as we heard yesterday, aniridia relates to an absence of the iris, but in many patients the iris is present, but it may be partially anomalous in different degrees, or the iris may essentially look normal. So although the disease is named about concerning its iris morphology, it's a panocular disorder, so it affects all the structures in the eye. The ocular features can occur at any age, from early childhood, teenage life into adulthood. And the major ocular features are corneal disease, glaucoma, cataract, iris defects, foveal and optic nerve hyperplasia. And patients usually have poor vision in the nystagmus related to foveal hyperplasia, which you can see here with the blood vessels crossing the foveal area, or also on OCT, depending on the degree of nystagmus. So it's a condition that affects the whole eye. And if you look at mouse models of aniridia, this is a wild-type mouse. So you have some labeled limbal stem cells in blue and white, and you can see the centripetal movement of the stem cells. And crossed onto a mutant mouse with a PAX6 mutation, you can see that this mechanism is disrupted. And so the corneal defects appear in development, so you get a reduction in corneal thickness, but then in adulthood, you get a limbal stem cell deficiency in the mouse. So the defects in the mouse reflect either limbal stem cell deficiency or an increased corneal epithelial cell loss. And we see this in humans. We discussed this yesterday. So here we can see pannus, neovascularization, corneal opacity, and then this conjunctivalization of the corneal surface with fluorescein uptake. So patients develop a progressive corneal scarring with age, usually in their teenage years. So the treatment for aniridic keratopathy is challenging. So you can use reduction of light, so tinted lenses or photochromatic lenses, preservative-free lubricants, autologous or allogenic serum, amniotic membrane. We discussed yesterday reconstruction of the limbal stem cell population, and then potentially subsequent PK or keratoprosthesis. But all of these maneuvers usually require intensive immunosuppression and have significant clinical challenges. So I think sometimes in medicine, prevention may be better than cure, and that's the sort of concept that I have in my mind. So if we go back to aniridia, so it's a dominant disease normally, 2/3 of cases, due to mutations in PAX6. In 1/3 of cases, it results from a chromosomal deletion, and the importance here is that the PAX6 gene can be deleted, but also the Wilms tumor suppressor gene can be deleted. So in a sporadic patient with aniridia, they have to have screening for Wilms tumor with ultrasound or undergo a chromosomal analysis to determine whether there is a deletion here. So to explain the rest of the talk, I have to go back to university days. And so this is the central dogma of molecular biology. So we have a DNA, the gene, we have the message, the RNA, and then we have the protein. And these are the 3 steps to generate a normal protein. So for PAX6, this is the DNA. So we have mutations scattered across PAX6, and most of the mutations form stop codons. And there are also multiple mutations. So if you want to develop a gene-based therapy, it has to address the genetic heterogeneity of the disease. So the body deals with these termination or stop codons with a process called nonsense-mediated decay. So if the gene has got a stop codon within it, the body has a system to prevent that RNA from making a protein. And so this is the mechanism of aniridia. So here is a normal person. You have 1 copy of your genes from your mother, 1 from your father, and so you will make 2 functional proteins. In aniridia, you have 1 copy from a wild-type copy, which makes a normal protein, and then you have a mutant copy, which fails to make a protein. So basically, you have 50% of the protein level that you require for function, and we call this haploinsufficiency. So one way to override that nonsense mediated decay or to stop the production of protein. So here's a normal messenger RNA being made into a protein. And when you've got this stop codon, it doesn't get made into a protein. So there are drugs that will prevent nonsense mediated decay. And one of them is called atalurin, and it's been used in cystic fibrosis and Duchenne muscular dystrophy. And there was a very interesting paper 2 years ago in which this drug was used in mouse models of aniridia with good effect. And so that's one approach to prevent haploinsufficiency and normalize the protein levels of PAX6. But I've been thinking about a different way to do that, and that involves the use of microRNAs. And microRNAs are small RNA molecules which control our protein levels. So here we've got the DNA, and it forms a messenger RNA, which normally makes a protein. That was that central dogma of molecular biology. But the microRNA gene can control protein levels. It can bind to the messenger RNA and affect the level of protein. So the way I sort of describe this to people is we all get flights all around the world these days. So these are the cities. So this could be Rome, New York, London, Sao Paulo. And the microRNAs are like the aircraft that fly around the world to all these different airports, and they can switch on the different airports or turn them off. So it's a very complex network of microRNA regulation of genes and proteins. So what happens is a microRNA, here's a gene, it binds to the messenger RNA, and it prevents that messenger RNA from being made into a protein. So this is the PAX6 gene, and this is its 3' untranslated region. And you can see using bioinformatics that we can predict a number of microRNAs that regulate PAX6. So the idea is that we use these microRNAs to try and prevent this degradation of the normal copy. So we do that using a target site blocker. So here we have the microRNA binding a gene, preventing its translation. But if we use a target site blocker, which will compete for that binding site, you can rescue the expression of that gene. And so we've done that for normal corneal epithelial cells. So here's the control cells. This is the expression of PAX6. Here's a mock transfection. And if we transfect with a target site blocker, you can see that we can we can increase significantly the expression of PAX6. And we can also do that at the protein level. So using this approach, we can upregulate the normal copy of the PAX6 gene, hopefully normalize the levels of PAX6 in cells, and that would form a mutation-independent gene therapy for aniridia. So to develop this, we've set up a consortium of patients that involve Aniridia UK and Aniridia Europe. And we have a team of scientists and ophthalmologists based in the UK, Paris, Israel. And really we'd like to develop a European consortium, and that may be something that SICSSO can help with, so that doctors can contribute patients and clinical data so that we could establish a clinical and research network for aniridia-related keratopathy. So thank you for your attention.